Good morning all, I am Kavya MC, Assistant Professor from SNS College of Pharmacy and Health Sciences. Today we are going to discuss about biomolecules. Biomolecules are nothing but those molecules that are found inside a human body or any living organisms. They can be examples can include carbohydrate, lipids, protein, nucleic acid, etc. One of the major biomolecules that are present inside our body is carbohydrate. Carbohydrates are nothing but polyhydroxyaldehydes or polyhydroxyketones, or the compounds which produce polyhydroxyaldehydes or polyhydroxyketones on hydrolysis. The molecular formula includes CnH2ON, where N is the number of sugar molecules. So some of the examples includes in carbohydrates are banana, potatoes, rice, meals, etc. So main function of carbohydrate is the quick release of energy, storage of energy, as well as giving structure to cell wall in plants. Some of the examples are sugar, starch, etc. Coming into the classification of carbohydrates, it can be classified into monosaccharide, disaccharide, oligosaccharide and polysaccharide. Monosaccharide having only one number of sugar unit, disaccharide having two numbers of sugar units, trisaccharide to nanosaccharides includes the number of n is equal to 3 to 9. So it is totally termed as oligosaccharides, and another last classification is polysaccharides. Coming deep into the glucose chemical nature, it can be expressed in different forms: Fischer projection formula, Haworth projection formula and Cheyer formula. Coming into Fischer projection formula, first one is Fischer projection formula is the representation of glucose in straight chain form. So it can be optical isomers. Optical isomers means when a beam of plain polarized light is passed to a carbohydrate solution, it will rotate the light either to right side or the light will rotate to left side. If the light is rotating to right side, then it is dextrorotatory, or if the plain polarized light is rotating to left side, it is levorotatory. So here we can see the example. If it is dextrorotatory, then the OH functional group will be present on the right side, and if it is levorotatory, then the OH functional group will be present on the left side. Enantiomers is the second classification. Enantiomers are the mirror images of each other. So here also we can represent it as L-glucose as well as D-glucose. D-glucose, in which the second anomeric carbon in which the hydroxyl group has been attached will be on the right hand side of a person. Similarly, OH group is on the left hand side of the person if it is L-glucose. Next comes epimers. Epimers are those monosaccharides which differ from each other only in the configuration around a single specific carbon atom. If it is in C4, then examples includes glucose and galactose. So glucose is having CH on left hand side on fourth carbon atom and OH on right side on fourth carbon atom. So glucose and galactose, if you are searching on galactose, then it will be vice versa. So glucose and galactose are epimers. Similarly, if you are considering the second carbon atom, glucose and mannose are the epimers because they have the different orientation in the second carbon atom. Next classification is Haworth projection formula. It includes the closed form. First one was the straight chain form. Here it will be the closed form. In closed form, it can be pyranose form or furanose form. If it is 6-membered ring, then it is pyranose, and if it is a 5-membered ring, then it is furanose. It involves anomers. Anomers are those they differ from each other in the configuration only in anomeric carbon. Anomeric carbon is nothing but the C1. So it can be two type, alpha as well as beta. In alpha, the OH will be below the plane, and in beta, the OH will be above the plane. So if this anomers of D-glucose interconvert in aqueous solution by a process that is known as mutarotation. So this is an example for mutarotation. The alpha form 36% and beta form 64% will undergo changes from interconversion from one form to another form if it has been mixed into an equilibrium mixture. Next is tautomerism or analyzation. Here also it will be having two different structural isomers that are indicating that this compound exists at mixture of two isomers that are equilibrium with each other. You can see the examples. Shifting of hydrogen atom from one carbon atom to another results in anadiols. Examples are glucose, fructose and mannose. Last one is chair form. In chair form, this can be represented as strain-free chair configuration in 6-membered form in pyranose. So next is the biological importance of carbohydrates that we will discuss in the next class. Thank you.